Hey everybody, how's it going? I wanted to do a little drawing here on my whiteboard today for a video for our lesson today. First, before I start that, I wanted to say that I read through everyone's questions about cancer and notes about cancer that they took, and while I gave a lot of people individual feedback on that, a lot of those questions I left unanswered because we will be covering them in this lesson and over a couple lessons next week. The video for today is going to focus on the role of DNA and genetics, and setting some background knowledge for some of you. Now, some of you have come right out of freshman bio, understanding this. Some of you have taken AP bio, so this is going to be a review for some of you, but for some of you, it's going to be new. The question is here, to understand the role of DNA and cancer formation, what does DNA do? Everyone should recognize this ladder shape, or this twisted ladder shape, I should maybe draw like this, of the double helix of DNA in the nucleus of all our cells. So I'm just going to keep it simple like this. DNA is in the nucleus of the cell, and what does it do? I'm going to keep this very simple and to the point. DNA is coded instructions for making a protein. And here is, let's call it protein X, right? DNA, instructions for building proteins. Right? Now, what do proteins do? The quick answer is proteins do almost all functions of cells. There is very rare a time when we see a catalytic function, a function that it causes a reaction in a cell that's not involved with some sort of protein. We have close to 200,000 unique proteins in our bodies. And each one of those proteins is encoded by a specific piece of DNA, a gene, for that protein, right? I should add a side note here that not all DNA is a coding region. Only genes are, but again, we're trying to keep this on the simple side. The coding parts of DNA, what do they code for? They're instructions for making proteins. Proteins then do functions, right? So I'll write the word function here. The protein's going to perform a role in the cell. So what could that function be? Well, it could be a protein channel. You probably remember those little gateways in the cell membranes where things go in and out of the cell, like we saw on some of our neuroscience lessons. The protein could be neurotransmitter is not a great example because neurotransmitters are very tiny, and so they would be really, really tiny proteins, or as you remember, probably amino acids. But a very simple protein could be a neurotransmitter, could be a hormone. Proteins are antibodies fighting disease. Proteins do a whole range of functions. And one of the things that proteins do is they control the cell cycle, right? So remember this drawing here. I'm just going to quickly draw the cell cycle diagram. Again, this circle here is not a cell itself. It's just a diagram, a little pie chart diagram. We've got our G1 phase. We've got our S, our G2, and our M phase. This is our cell division, where the cell divides. You will remember from previous lessons that there is a G1 checkpoint, right? There is a G2 checkpoint and an M checkpoint. These are parts. These are parts of the cell cycle where certain proteins, I'm using that word now, proteins need to be present in order to pass the checkpoint. So there's two things that I want you to add to your notes today, and that is tumor suppressor genes, right, and oncogenes. Tumor suppressor genes are proteins that are genes that code for proteins which halt the cell cycle. All right, that's it. So if there's a protein, a tumor suppressor protein, oh my God, my marker's dripping, that becomes present at a certain time in the cell cycle, it's going to stop that cell cycle. And you've learned a little bit about, I think, G0 phase, where the cell can be halted. It's like the brakes on a car. If you can understand brakes on a car, you can understand tumor suppressor genes, right? That the protein gets produced saying, stop, right? Oncogenes are another form of quote unquote cancer gene. Oncogenes are like the gas pedal. Oncogenes, or more accurately, proto-oncogenes, become proteins. They encode proteins that push the cell past checkpoints. So just to sum up, if we get to a checkpoint and a protein is produced to stop the cell from dividing or from continuing its cycle, that is a tumor suppressor gene, right? It stops cell division. If we get to a checkpoint and there are proteins present that move the cell past the checkpoint to keep it going, those would be proto-oncogenes. Now, there's no. I'm gonna take this actually this off and kind of look at you here for a second. There's no such thing as cancer genes, right? There's no gene that some people have that others don't that causes cancer. However, what we do have is we have healthy genes that halt the cell cycle, tumor suppressor genes, and we have healthy genes that push the cell past cell cycle checkpoints. Those are proto-oncogenes. When those genes become mutated, or if they become mutated in some individuals, meaning the DNA changes slightly, it can produce a faulty protein, right? So if it is a tumor suppressor gene, where there's a mutation that causes the protein to be the wrong shape and therefore do the wrong job, well, now we've just lost the brakes on our car. If it's a proto-oncogene, again, that's O-N-C-O, oncogene. Proto-oncogene, that's a healthy gene that pushes the cell past checkpoints. Let's say that that gene gets mutated in a way, and this is an important point, that gene gets mutated in a way where the protein that encodes is constantly active, right? We don't want the gas pedal pushed down all the time in a car. We want to ease off the gas at times to go the right speed. Same with proteins. They get turned on, they get turned off. Let's say there's a mutation in a proto-oncogene that makes a protein that's supposed to push the cell past this checkpoint and then turn off. Let's say that protein stays active, and now the cell is just flying past checkpoints again and again, dividing. That's like a broken gas pedal on the car, and that can lead also to tumor formation. So again, I know I've done a couple summaries here, but just to sum it up one more time, tumor suppressor genes are like the brake pedals on a car. When mutated, they're like broken brake pedals. The cell doesn't stop, doesn't get that halt signal, the stop signal. Proto-oncogenes, healthy genes that produce proteins at the right time to push cell past checkpoints. If there's a mutation there that causes that protein to be constantly active when it shouldn't be, now the cell is passing checkpoints. That's the broken gas pedal. Okay? So genes make proteins. Proteins do functions. If the function is in relation to the cell cycle and there's a problem in that function, now we can have an understanding of how our genetics can be more likely to lead to cancer than others.